Good afternoon. My name is Douglas Drake with The Edge. I want to welcome you to our first, actually, in a series of webinars using the BioRails platform with Morphit, which is an extensive platform for any type of biological sample management, assay management, and so we are actually going to be illustrating how this can be used for a variety of different drug discovery processes, both from genomics all the way to early safety and efficacy. But today we're focused on the analytics for DMPK and ADME, so I appreciate you joining us. I will introduce our speaker shortly, and so then without further ado, let me introduce Dr. Andrew Lemon, who's the CEO of The Edge, who will actually do the company introduction and then introduce our second speaker. Thank you very much.
Okay, thank you, Doug. I'm very excited for us to talk to you about the data analytics for DMPK, and we're going to review some of the challenges involved with delivery of analytics solutions into the DMPK environment. Peter will demonstrate to you the system, and then finally I'll round up with some of the benefits, and we'll take any questions at the end. Okay, first of all, I thought a quick brief introduction to The Edge. So The Edge are an independent company. We're over 10 years old. We specialize in providing solutions for the biopharmaceutical and pharmaceutical research areas. Our software is used by 4 of the top 10 pharmaceutical companies and many of the medium tier companies across the US, Europe, and also the APAC region. We deliver solutions across a number of different areas, as Doug mentioned during the introduction, covering everything from global assay requesting and planning solutions to data management in areas like DMPK, ADME, in vivo pharmacology and other in vivo areas, screening and biotherapeutics. We also provide modules for compound management, supporting ordering and delivering of inventory. So really covering the full workflow for a biologist from demand generation all the way through to inventory, supply, and testing. If there's one thing we're passionate about, it's helping people to optimize their processes and also keep the value and structure of their data. And that's really what drives us. So today we're going to go through and have a specific look at our data analytics module for DMPK. We're going to review some of the challenges that DMPK and ADME pose for delivering solutions. And then we'll actually see the system in action, and finally I'll review the benefits before we take your questions. So a little bit about those challenges. Well, there's 4 main areas which make delivering solutions into the in vivo and in vitro DMPK environment particularly challenging. One is in terms of variability. The second is the flexibility required to cover a number of different conditions and types of assay that are being operated in that environment. Also to meet the workflow requirements for scientists working in those disciplines. It's often not a single person activity, it's using teams of different people, whether it's in vivo, through to bioanalysis and back, or other disciplines as well. And of course also the challenges of reporting that data. These are the types of area and assays that we're dealing with. Today's webinar is going to focus particularly on in vitro drug metabolism support, and a later webinar will cover the in vivo area. So we're going to talk about assays in terms of areas like permeability, distribution, stability, binding, and interaction areas. And we'll cover those different types of assays and talk about some of the challenges of delivering analytics into those environments and how they change over time. If we talk about variability, we're talking about a variety of different mass spec instruments that are being used across different laboratories, which may have subtle variations in output. They'll have different file formats. They'll be operating with different plate formats and plate sizes that need to be adapted by the software. And here's a simple example of an output file from the Waters Analyst software. You may have different sample identifiers, different species and strains in here that need to be accommodated by the software. We also need to deliver software that's flexible enough that it can cope with variations in the number of samples that are being tested, the plate formats and sizes, different species that you're operating on, and also other variability in the parameters that are used to process those observations and results. There's a couple of examples in here. So it might be scaling up from a 96 well plate to a 1536 format. Also, we have areas like scaling factors. So particularly for CROs, they may have multiple clients which operate multiple species for their assays, and they're going to need to automatically look up the scaling factors which are pertinent to those particular combinations of client and species in order to get the results those clients need. Another challenge is managing the workflow. So obviously we've got to manage everything from receiving samples to tests, through tracking their inventory requirements, maybe ordering plates to be delivered, through the plate preparation process, and allowing you to efficiently process work lists, and also interact with different teams, whether it's an in vivo scientist dosing an animal and then taking biosamples that need to be analyzed bioanalysis, or it could be formulation of a compound ready to be used in an assay. Not only do we manage that macro level workflow, we're also looking at the micro level workflow within the lab itself. So here would be perhaps a typical workflow. So we're going to prepare the plates, we're going to incubate the microsomes, going to add the compound, then sample the wells, quench those plates, and then obviously run them through the mass spec, and then bring everything together to do the analysis. So what we've done is to create a solution that allows you to manage all that data analytics environment. It comes with something called Dimensionality. That basically means it expands to meet the needs of your data. So if you switch between a 96 well plate to a 384 well plate, all of the analysis capabilities will just expand to meet that extra data. Or whether it was an extra treatment group in an in vivo study, or whether it be an extra species that you're testing. The analytics environment comes with all the data analytics you need to operate those assays. So it comes with inbuilt curve fitting engine, graphing and charting, statistical analysis, and also non-compartmental modeling for PK. The system provides an electronic protocol which helps to guide the user of the system through how to operate the analysis and the experiment. The same analytics solution is provided into a number of different flexible deployment environments, which I'll outline. So let's talk about the first aspect here, which is the elasticity of these templates. What it means is they can expand to meet the needs of your data. So they can have groups and subgroups. If there are different dimensionality to your experiment, as I mentioned earlier on, such as plate size or number of treatment groups, they will expand to meet that data. They also provide the ability to be able to make relationships between the data, so you just get the available calculations and parameters that are available to that particular set of conditions. It's all model-based, so all the calculations will happen automatically, and it understands your scientific data files, so you can bring in data from reader files, from mass specs, from fluorescence instruments, etc., and immediately use them into the analysis environment. As I mentioned, it's got built-in analytics capabilities. So you could do curve fitting for things like IC50, EC50s. You can do charting and graphing of data with interactivity. There is statistical support, so we can calculate statistics of analysis of variance, etc. You can define your own calculations, so really you can adapt this system to meet any type of assay. As I mentioned, it's all instruction driven, so there are a simple set of steps that you can follow through the instruction sheet to drive your analysis. That makes it easy to learn for end users using the system, so it guides them through how to run their analysis and process the data. This same data analytics environment is provided into a number of different enterprise platforms. First of all, there's our own BioRails enterprise platform that provides you with support for the full request, schedule, and execution workflow. So you can request assays, you can schedule them, you can run the assays, you can do the analytics, you can access your data either directly through third-party tools for querying or through web services for data access. So it's fully compatible with all modern reporting tools. It provides lots of integration options. However, we also, through our partnership with BioVia, provide the same data analytics environment into the BioVia ELN as an embedded section. This means it can be used for GLP experiments, where we feed the audit trail from BioRails, which is a fully functional cell-by-cell audit, in with the BioVia section type audit. You can also use this as a pure desktop solution for personal productivity. That's useful into environments where we're doing sort of personal work to work up data. Also, of course, it can be used with these different enterprise platforms. So at this point, I'm going to hand over to Peter Vine. Peter is our senior consultant and data scientist, and he's been working with a number of different clients to build these assay templates and build this collection that we're now talking about during this webinar. Today, we're going to cover most of the in vitro areas with example data analytics templates. We won't be covering specifically in vivo pharmacokinetics today, but it will be the subject of a future webinar. So with that, I will hand over to Peter.
Thanks, Andrew. As Andrew described, I will start with opening Morphit as in the desktop environment. So it's a pleasure to be able to demonstrate some of our tools to you. So the only thing I need to type in here is Morphit, and then it will just open the desktop application. So let's just open a new one of the templates that we have built for this specific purpose of the DMPK support. So I will start with opening the brain binding, the brain tissue binding template. So it's nicely organized, as you saw, in these different categories that will be just stored on your hard disk or on a shared drive. And it opens as this. So this is typical for the binders we build. We make you land on the landing page. It has a fixed structure so that it is easy for you to recognize one of the binders in this specific package. The only thing basically that you need to know of is these instructions, and you follow these instructions from 1 to 8 in this case. So let's just press this button and start with importing some data into this binder. So this will open just this dialog window where you get access to any file on the system. And what we are going to open, of course, is a brain binding mass spec data. So this comes from a Waters instrument, and I can briefly show you how it looks like. So I will open this with Notepad first, and then you will recognize, I hope this is familiar to you, this is a typical plate reader file as it comes out of the mass spec instrument, the analyst software. It's containing details about the compounds and all that. It's basically all organized, but many of these files, although they seem very similar, they can have missing columns, they can have different information in all these columns. So Morphit is a very flexible tool to read this all. I double click on the file, and now Morphit will open this Excel file or this text file. First data it will present is just the raw data. So the next step to do is instruction number 2, and then what happens in these files is, although many conditions are captured in the file itself, sometimes there are experimental conditions that need to be filled by hand on top of it. So that is when we build a button like this. And then what this binder needs is it has almost everything except the initial substrate concentration of the brain, homogeneous in this case. So which is set to 10, but you can change that. So you can give it a default value, and I switch it to 10 again. So that is just combined with the other data to work out all the analyses. So then we typically move into the inspection part. And for the purpose of quality control, you can just inspect your internal standards, and that is done in this sheet. So here you see a couple of numeric fields. This is the area of the mean, or the mean of the area in all the internal standards, and the standard deviation. So you could see in one go how they are distributed. So there's 203 measurements of the internal standards in this file. The coefficient of variations is 23%. And you can judge for yourself here on the graph that there is a tendency for the internal standards to go up here and then remain constant. But this is pretty much typical for what we see in these files. So if this is all over the place, you could decide to remove the internal standards by using this tick box, and then instead of normalization with the internal standard, the raw data will be used instead. So this is an example where you have a lot of control already at the onset of working with the binder. But next comes the most important, the central part of the binder, which is the interaction with the data. So if you press this button, you see the core of your data. So all the data is nicely organized. Here you see the batch that you are testing, your tissue concentration, and then you work your way to the right, and then suddenly you see 3 replicates of this compound. So this is automatically recognized in the file and organized by Morphic. So if there would be 6 replicates on the plate, this will just expand to 6. That is what Andrew meant with by the auto-dimensionality of Morphic. So, and each of these replicates, of course, has a donor concentration and acceptor concentrations. So you can also tick boxes like this, where yeah, I can explain very briefly what you can do with all these data, because the first thing to do is you can knock out the data value by hand, or by a quick key like Control K will just knock out this single data point manually. So if I knock out this data point, all the related values, not only in this table, but also in all the linked tables, will automatically be knocked out. This will all be audited, by the way, as well. So the second way to interact with your sheet is to click tick boxes like this. So in case the acceptor is larger than the donor, which in principle cannot happen, of course, but due to measurements errors that can happen, it will be flagged up by the formula. As you see, the formula just detects if the acceptor is larger than the donor, but still you have manual control over it as well. And then it flags it up as orange and it knocks it out as well. So this is what we typically do if there are tests or quality needs that you have to control. So we build a couple of these tick boxes. And then on the far right, you see the aggregated functions applied to the individuals. So these are typically averages, they are counts or standard deviations across these. So, and again, Morphic will compute this correctly, even if you have got 8 or 10 or whatever number of replicates. There is no need to expand it yourself. Morphic will take care of that itself. So, and then there is a comments box, and we have pre-populated it for you. So these are the typical things you would like to comment. So these are all pre-filled from other tables, and we can just take your preferences and stick it in there. And on top of that, you've got your recovered fractions and all that. So it's pretty much to look at, but you are in total control. This is exactly showing you what the type of control that you want to have on an individual replicate level. So the result of that will be this very, very simple table. And suddenly it becomes very easy to explain. Here you've got your batch. So this is the output of the previous table. It's populating this table. You get your unbound, your bound fraction, your standard deviation, the corrected unbound. So this is corrected. You can see that on the formula as well. That is correcting for the dilution. So this is the percentage recovered. It must be close to 100%. And here's the recovered SD. And here you see the comment I put in automatically. So this can also be presented as charts. Not everybody appreciates having charts. Some people are it's just sufficient to have the tables. But all the data is plotted as charts as well. So here you get the fraction unbound with your standard deviation on it. So the final step is to export this data table to a database. So in an integrated environment, we could do that by just pressing this button and then consider it done. All the data will be in the relational database. And then even the binder itself will be stored as a digital object in the database as well. Morphic offers you excellent ways to export it to other applications. So then you just press File Export, like you can do in Excel, and then you can pump data to a PDF, including all the pictures, to Microsoft Excel. So again, PDF, CSV, Excel, XML, HTML, and of course the database BioRails itself, if you're working in an integrated environment. I'm going to open a second binder. So I click on File, New, and then I move on to another template. So we've had the binding, an example of the brain binding. There's also plasma binding, and we've got more for you. But now we move to the distribution kind of binders. And we've got this blood plasma distribution assay. So this assay is aimed at studying the distribution between red blood cells and the plasma. And obviously it is a pretty important predictor for how much drug you have available in the bloodstream. So it has pretty much the same structure as the other binder I showed you. So here are a very limited number of instructions, and let's just push the first one. So in this case it is asking for an XLS file, an Excel file, and we double click and Morphit will open that particular file and show it to you. So this is again the raw data to make sure that everything was read. So this is a pretty long table, and it's color coded. So that's all fine, but it's not worth looking at it because we immediately dive into the interaction. That's the second one. So again, you have all the options here to your service. And as you see, Morphit has nicely organized all the data into groups of species. So this sample was measured in the mouse. It recognizes that from the species column in the file, and these columns were all recorded in the red. And then from there onwards, for each sample was measured in the blood and the plasma matrix. So if you would have the need for a third compartment, Morphit would just scale to that. So in this case, it's a 2-compartmental analysis, but there is no limit to that. And each blood and plasma matrix part is done on 3 replicates in the file as well. So you will find them here. And then you get your relative response. So this is normalized by your internal standard, and you get nicely the 3 values. Again, you got all the options to knock it out. So Control K will knock the value out. And then you see that in a little yellow cross here, because in this little micro chart, all the data is displayed. This is a nice example of where you can knock out directly in the graph as well. So this blue dot here might be an outlier. It's for me to judge, and if I click on it, then it should just knock it out. So you can do the same knocking out as the manual knockout on the numeric cell. You can do that on the graph as well. And immediately the average will just adjust. So this is the blood plasma ratio and some other measures. So this is the upper and the lower confidence band, and that will all be pumped into the results table. So this is the nice and simple output of the assay, where you just get your sample, your species. So these are a couple of controls here, by the way, midazolam and verapamil, and then you get your response and the ratio. And that can then, of course, be shown as a chart. In this case, it's just one simple chart where you see all the blood plasma ratios in just one big graph. All these binders are organized in one and the same way. So there's 5 in total that I'm going to show you, and we now will move into the interaction assays. So we now move in the cytochrome P450 system. In this case, there's 2 variations. So this is just the percentage inhibition, the single point measurement basically, and this is the IC50 measured at multiple concentrations, and then you get an IC50 out of it. So let's open the complex one, and this will now open quite a big binder that will read an Excel file again. So it's your landing again on the landing page, so this starts to become familiar, I hope. And then we first start by importing a fluorescence data set. So in this case, it's not a mass spec, but it's a fluorescent data file. And I can very briefly open that. Let me see if I have the correct one. So it's this one, the IC50 interaction file samples. And I open this with Excel first. And then you see this is typical for a fluorescence reader, just the plate data. And here you've got the different enzyme systems as recorded on different sheets. So this is the way we got the file from the customer. We didn't do anything to it. And all they were asking, okay, read the raw data and get yourself organized with it. So that is what the binder does. So I double click on the file, Morphit will bring the data in. So here it is. So that's a truly long table. So we can now move on to the process data. And again, I have now opened just one file, but if your data happens to sit in multiple files, Morphit will deal with that correctly. You just select multiple files and import them at once. So here we are. This is the organized output, and the instruction told the binder to move into to straightly jump into the calibrations part for quality control again. I can just see the highs and the lows on the plate in a nice way, and also depicted here as a graph. And also some averages were done on it, so there's 8 replicates of the fluorescence at a high value and all that. You get some C prime scores as well, so to judge whether you are in the right range. And we can even set some rules to constrain your data and to knock it out automatically. So the next, of course, is the interaction screen, which you get a complete overview of everything. So here's your inhibition computed. This is your fluorescence. This is your log concentration. These are all your replicates, basically. And from these data, the individual curve fits are also displayed. So on top of the population fit, which you see here on the right, so here you get your one fit through all the replicates, you can also judge if the individual points are correctly fitted. This is a 4-parameter fit in this case, so this is making use of the built-in fit capabilities of Morphit, which you can easily compare to the GraphPad Prism capabilities. So you can set the starting parameters, you can choose the model and all that. And you can also see that some of these tick boxes have automatically been ticked, and that has some consequences for the data processing as well. So that's one of the tricks we have to judge if your data is in the right direction. And this is the result from this assay. And here from on the left side you see all the samples that were tested, and here you see your enzyme system. So this is the 1A2. Then we move to the right and you see the 2C19, the 2C9 and the 2D6. And they're all separated by these little gray lines, so it's relatively easy to see the blocks in this format. And you can see that they're all color-coded to give you some feedback on what the drug is doing on that enzyme system. So all these enzyme systems are tested at once, and also you see that all the white space in between is inconclusive. Of course, you can also export this data back to the database, and that finishes the presentation of this assay. So that leaves us 2 assays still to go. So I will close this one, right, and move on to the next assay, which shouldn't be a big surprise to you. That is quite an important assay, which is the permeability assay. It's KCO2, which is a very wide standard. It's a widely accepted assay to estimate the human intestinal permeability and to investigate the drug efflux to predict the suitability of a drug for oral dosing. So it lands on the landing page again. And here you see a graphical picture of the experiment itself. It starts again by importing the file. So here we are. Let me see. Here's the KCO2 file. And in this case, it's loading up an Excel file again. So it's the same recipe, importing the file, see what the data is all about. So this is imported straight from Excel. It's a long skinny table, basically. And now we move straight on to the calibrations. So in this case, the data also consisted all the standards and the calibration lines. But this is a way that Morphit can still access these data, and you can judge for yourself if your calibration is proper. And in this case, you see quite some deviations at the low concentrations for this compound. So then you can inspect your internal standards. So we've been here with another assay as well. It's nicely distributed all around this sort of value. So if we are happy, we can just proceed. Inspect the start concentrations. So these starting concentration of these 3 points might be a little bit high because they are supposed to be in this sort of area. You could already decide to knock a few data points out at this phase. But if we don't do that, we just we move on to the next instruction. And this is quite an interesting one because it shows you in this particular lab where they use this setup, they also measured, they had an additive, luciferase yellow, to inspect if the permeability membrane was leaking. So they had another file format brought in from a fluorescence machine. So this is a completely different format, which is shown here. This is just the raw data. But once I press the membrane integrity button here, the next instruction, you see that it is concluding if there is a leak in these directions, so in these experimental wells. And it does that, of course, for every sample. I can just simulate a leak here. I can manually control these boxes if I want. We can also disable that, of course, that it is purely automatic. But we can decide, okay, this is so close to a leakage that I will mark it as a leak. And then we can interact with our data. And then you get this busy sheet again, or the busy table. And then in the yellow part, you see all the conditions. So these are the times of measurements. This is the direction. Here you see the analyte that is measured. And here you see all the standard concentrations. So everything is combined into one big table. So all the data is condensed as well. So here you get the averages, the C0 concentrations, the apparent permeability, the recovery of the compound. And it's all under control of these tick boxes and the knockout methodology that Morphic is supporting. So if you're happy with all of this, then you can just move on to the next table. And that gives you the nice two-dimensional simple structure where you've got your conditions here as a string. It's easy to read. These are all your conditions extracted from the settings. Here you've got your analyte, the project, the apparent permeability operator. So this you can also have a smaller than or a larger than. The binder will conclude that for you and will use the operator in this column. Here you've got the values. It's all color-coded so you can immediately judge which are high and which are low. And then the extraction ratios and the absorption. So these two columns here are interpretations based on some business rules. So we've got low, medium, high categories, and also here a verbal expression of what is happening. So all automatically done. Here we have the apparent permeability. Here is the efflux ratio, and here is the recovery of the compound, which obvious problems here with this first compound. This should be around 100%, of course. And that concludes the KCO2 binder. So this nicely shows that Morphic is able to combine several data sources at once. It organizes the whole lot, and it does combined computations on all of them. And then I move to the stability assays. And these are the hepatocytes, the microsomal, and the plasma stability assays. And of the three, the hepatocyte might be the nicest to demonstrate to you, because that also shows what Andrew has called the combination with the lookup data to predict data in human. So it starts, in this case, with a settings instruction. So I press that, and here you can enter the cell density, your sample concentration, and the minimum area that you want to achieve in order to make a valid reading from the instrument. So then we press the import, and we bring in the hepatocyte stability data from the MS instrument. So here we are. So then you can provide some additional sample info, and in this case, as you can see, every drug was measured in 2 repeats at different concentrations. So you could say, okay, I've measured at 3 and 2, for instance. You could change this. This is manual input. But this is the default as it picks it up from the settings. So then we move on. We can have calibrations. So there are no calibrations in this file. Then we can interact with the data. Here you see the linear decay model that is needed for the decay of the hepatocyte activity. And here you see the linear decay of the compound, and here you see the control that was done simultaneously on the plate. Some labs use this method, where you use the log of the remaining drug on the y-axis versus time, and then you can do a linear decay, of course. But some labs use this method, where you have the linear concentration or the linear remaining compounds put on the y-axis, and then, of course, you have to do an exponential decay model. So, in fact, these two are pretty compatible, but statistically there is a difference. And that is just because of the variance of this is homoscedastic, where you have equal variances at every time point. Whereas in this model, it varies. The variance will be larger at this high point than here. So this is often the preferred model when you talk to a statistician. But nevertheless, it could well be that you have a preference for this, for instance, because your R squared is larger. You see in this case they are equal. In this case it's slightly larger, then you prefer this one. So you have full control whether to use this or this model. And we have built in this tick box for that. So if I click this tick box, then it will automatically select this model. And as you see, while I am clicking this, you will see these values changing. You see? So it's slightly changing here from 0.04 to 0.06, so it affects your data. Of course, if you came to a conclusion that this is the only method your lab will support, then we will just get rid of this one for you. It's just disabling it or take it out completely. Just one table to go, but this is a nice review of all the data where all the concentrations are put in combination. Where you see that the decay is dependent on your initial concentration, as you see here. So this is measured at 3 mg per mil, and this is measured at 1 mg per mil, and that is now combined into this graph. So that is just for your comfort. We are combining all these graphs into this table, and then we push it through to the results table, which is this one. And there you have the samples, of course. It's nice and flat. You get your concentration here, your intrinsic clearance, and then these are quite interesting because these are the scaled CLs and the predicted clearances in the human. This is done by having values for the liver blood flow, the liver weight, the microsomal protein, and the hepatocellularity of that specific species. So this table contains all that data, and as you can see, it can also be made project dependent. So if you're working for a CRO and you have to use data for different customers, then you just have to add one table, and in this case it's all containing the same data, but these can be different. The result table is always using the correct table based on the project for this compound. So that is all happening under the hood of Morphit. So this concludes my demonstration of Morphit for a series of DMPK binders. Thanks.
Thank you, Peter, for that very comprehensive review of those binders, which I hope everybody found interesting. So some of the benefits of this, first of all of these binders that we've shown you, they're all come with full documentation. They've all been through quality control, so they're clear and understandable, they're robust. They're all workflow driven, so they're easy to use, so that training barriers are low. They've all been scientifically validated. We've developed a lot of these binders in collaboration with both customers from a pharmaceutical environment, such as Merck KGaA, and also with contract research organizations such as Evotec that are using these same binders. So we know they're dependable and reliable. They also have a standardized design, so as we showed during the presentation, so they're quick to implement. They have a consistent design, so they're easy to learn. Once you've learned one binder, it's easy to pick up others and see how they work. They're also adaptable, so you can adapt them to the details of your data analysis, and also you might adapt them to different file formats and plate layouts, etc. during their operation. You can operate them in standalone mode, as we just showed you, or they can be fully integrated with the BioRails platform or the BioVia ELN. Future plans in this area, we're investigating also building a collection of data analytics binders for in vitro toxicology, transporter assays, and also we've already built some examples of generic in vivo study models. If you think there are binders that you would expect to see in the DMPK collection that aren't there, we're very happy to work with you to either adapt the existing binders or add those new ones. We also have some very nice examples of for the in vivo PK, which will be the subject of a future webinar. Xenograph studies for oncology, some biotherapeutic examples, and genomics as well. So at this point, I'm going to hand back to Doug, and we're going to start going through your questions, but thank you for your attention.
Which assay design is implemented, which in PPB, for example, ultrafiltration or equilibrium dialysis, input raw data could be different?
Yeah, definitely. I can answer this question. In this case, the one I showed you was the equilibrium dialysis. Indeed, we can work with the other assays, and then it's indeed reading from a different raw data file. So sometimes there are some changes needed to the binder, which we can provide. So the next step, of course, is to build an ultrafiltration binder straight for you to use.
If I am using Morphid by itself, can I protect the results of my experiments so they cannot be changed? Yes, I would want to set the summary business rules myself, so I can set the text for each category of the result.
So there's a number of ways. So first of all, obviously, if you want long-term protection for the intellectual property on your binders, then the best way to do that is to use it in combination with an enterprise platform like BioRails, which has the electronic signatures and witnessing, which really is the long-term solution for protecting the data. You can, however, protect data in the sheet. So as you operate on the sheet, you can apply protection to certain tables and columns as you go through the operation of the binder as well. But our recommendation is, if you want to protect data, is you store it in the enterprise platform, you sign it off and witness it, so it's rendered read-only and is also tamper-proof.
The next question, can you monitor controls from different experiments?
Yes, so I've actually, I saw that question. I pulled up this binder here. So this is another example. This is a microsome stability binder. It's fully integrated with BioRails in this case. And what this binder does is it's a metrics binder. So having run a microsome study, what it does is it queries the historic data back from the database over all the previous runs of the same controls and standards across all the previous runs of the microsome assay against the same species, and it gives you this graph here which shows you where your data point lies in respect to all of the historic runs over the dates that it's plotted. So you can immediately see if the current point is outside of, say, 3 standard deviations of the mean, for example. And we can do that because all of the previous data has been stored in the structured database inside of BioRails and is available for querying. And that's the advantage of using this analytics environment with a structured data store like BioRails. So yes, you can fully take advantage of all your historic data here and query it and then bring that into the context of the current experiment.
The next question regarding the Z prime, can I store these values for looking at later?
Again, I guess that's really the same question here. Yes, you can. So if you're running a series of plate-based experiments and you're evaluating the quality controls over the plates and you're doing a large run, we have both automation tools which can process those plates automatically and also post you up a running monitor of the Z prime. And we can also, of course, store the Z prime in the database so you can then search and look for trends over time. That goes for both things like quality control like Z prime, but also the performance of controls and standards, so you can monitor long-term quality against your assays.
Well, I'd like to add that we have a number of actually previous presentations, and this presentation will also be made available, but we have a number of presentations, actually video presentations, as well as white papers and other things available on our website. Thank you very much and have a great day.